Okay, so this is a biology Throwback Thursday lesson, and what I need you guys to do is get out a sheet of paper, and what you're going to do is kind of keep all of your throwback notes in one area. So just a piece of paper is fine for the start of this today, and go ahead and date it October 3rd, 2019. Okay, so if you think back all the way to the beginning of school, we actually learned about two different types of cell plans: the prokaryotes versus the eukaryotes. Okay, so there's two basic cell plans on planet Earth. There's prokaryote and there's eukaryote. The prokaryotes, if you remember, do not have a nucleus. They do not have membrane-bound organelles. And they're pretty small, comparatively speaking, with eukaryotes. So there's no nucleus, there's no organelles, and they're pretty small. And an example are bacteria. Now when we talk about organelles here, what we're talking about are those mini organs with our, if you remember our organelle dance, the nucleus, the mitochondria, the Golgi, the lysosome, the vacuole, those are all organelles, and prokaryotes do not have them. Okay? Their DNA that's inside of a prokaryote is not in the nucleus. It's just floating inside. They also have things called ribosomes inside of them. They also have cytoplasm, and they have a cell membrane. So these things, the ribosomes, the cytoplasm, the cell membrane, and just the fact that they have genetic material are the things that they have in common with the other cell type. Okay? Now another thing they have too is a cell wall. Okay? And animal cells, for the other kind, the eukaryotes, do not have cell walls, but plants do. So cell walls are common between prokaryotes and plant cells, plant eukaryotic cells. Okay? So then let's talk about this other one, the eukaryotes. The eukaryotes. I'll go back to this green color, and I want to make it thick. Okay. So the eukaryotes are basically the antithesis of prokaryotes, or the opposite. They do have a nucleus, and inside the nucleus of eukaryotes is the DNA. That's the genetic material. It's inside. And if you remember, the nucleus has pores, little holes in it. And that's so the information from inside the nucleus, from the DNA, can leave. And so it does so with the help of its little friend RNA. And we'll get into that later, but you've heard that before. Eukaryotes also have membrane-bound organelles. Okay, organelles. I don't know if you ever noticed that the word organelles has the word organ in it. So these organelles are like mini organs. They have jobs, right? So there's the mitochondria, There's the nucleus, just to name a few, the Golgi. Okay, those are just examples of these complex organelles that the eukaryotes possess, and the prokaryotes do not. The eukaryotes tend to be larger. Make this bigger. So the eukaryotes tend to be larger than the prokaryotes. Eukaryotes can be single-celled or multicellular. Okay, so a single-celled eukaryote is something like an amoeba. Okay, and I'll show you like a little picture of an amoeba, or something like a paramecium. Okay, that's basically single-celled eukaryotic organisms. Okay. But there are also eukaryotic organisms that are multicellular. Okay, so the multicellular eukaryote examples would be humans, right? Because we have eukaryotic cells, but we have many of them, and that's why it's called multi. Dogs, plants, okay? And remember, plants have this in common with prokaryotes, the fact that they both have cell walls, okay? Fungi, which are like mushrooms. So fungi also have eukaryotic cells, okay? And then another simple thing to like kind of sum this all up with a term to make it like super simple is the fact that prokaryote cells are simple, whereas the eukaryote cells are complex. And the reason why they are considered complex is quite simply because of all of the things that are inside of a eukaryotic cell, all of those organelles. So what I want to do now is actually show you a picture of a prokaryotic cell versus a eukaryotic cell. And on the EOC, they will probably have a picture of prokaryotic cells and eukaryotic cells. And you'll have to identify then which is which. And so what I want you to get out of this here is the fact that this here is a very simple cell. I see some genetic material in there. And then when I look at this one comparative to it, you see all of these little organelles on the inside. There's just different shapes of things in here, like the ER, like the vesicle, the Golgi. These little red things or pink things here are mitochondria. And the biggest thing is the nucleus here. So it's very evident that this has a nucleus. So you want to be able to tell that difference between a prokaryotic cell and a eukaryotic cell simply by looking at it. Okay? All right, the next thing I want to talk about, I wanted to show you the paramecium and the amoeba. So let's go back. So a paramecium, they're really cool. Maybe there's a video of one. Let's look at this. It's pretty cool. And we don't need to hear it. So if you were to like take a sample of pond water and look at things in the pond water, you would see little life, single-celled life. So this is actually a paramecium. This paramecium has a nucleus and it has all these things on the inside. Those are all like the Golgi complex, the vacuole, the ER, all of that stuff. It has all of that inside. And this single-celled organism does everything that you and I do except it's in one cell. So it takes in energy and then it gives off waste. And so it has a mitochondria. It'll take in sugars. It'll break down those sugars in the mitochondria and turn it into ATP. And then it'll use that ATP. So it's basically the same as us. It's just, in one cell instead of multicellular like us. Okay? So we'll stop that. Another cool single-celled eukaryote is the amoeba. Amoeba is pretty cool. Amoebas are like kind of like your white blood cells. And here's a good one. It's hunting down a paramecium. So the amoeba is kind of like a killer whale of the pond single-celled life world. So here we have this globular thing is the amoeba. And the other day when I was explaining endocytosis, when cells bring things in, they wrap their membrane around it. You can see these paramecium have been wrapped up in the membrane of the amoeba, and now they're on the inside of the amoeba. So the paramecium is inside that amoeba right there. This is great footage. And soon enough, a lysosome will come over, right? And a lysosome will break that down and with the enzymes that it has in it from the Golgi that packaged it, and then backtrack to the ER, the rough ER that made the enzymes. And then from the ER, it got the information to make the enzyme from the RNA, which got the information from DNA. So what I just did there was I reversed the protein pathway, okay? So here again, you have these like pseudopodal extensions. These are extensions of their cytoplasm with membrane, and they're wrapping it around this single-celled organism, the paramecium, and eating it. Both of these both of these single-celled animals or protozoa are eukaryotic cells. Okay? All right. The next thing I want to talk about is the protein pathways. So now we will talk about the pathways. This is not working here. The pathways. So we did the prokaryote, eukaryote. Now we're going to talk about the pathways. The pathways, there's 3 of them that you have to know. And so the first pathway that I want to talk about is the protein pathway. And I'm going to put this in order here. Basically, there is the nucleus inside the eukaryotic cell. Okay, and in the nucleus, it has DNA. DNA is not allowed to leave the nucleus. So the nucleus has pores through which RNA can enter into the nucleus and get the message from DNA. So then RNA is like DNA's little buddy, and RNA will take the message from DNA and take it out of the nucleus. From the nucleus, it'll go to the rough ER, and the rough ER will convert or translate that message that RNA has into a protein. Now remember, enzymes are proteins. Okay, so it can convert it into an enzyme. Now the only problem is that this protein here, or enzyme, isn't packaged right. Okay, so it needs to get packaged and distributed. So then it gets sent to the Golgi. And the Golgi, I'm just going to put, will package and distribute, P and D, and then send the protein where it's needed. Okay, so that's the protein pathway. And you can pause this when you need to. Okay, the other pathway I want to talk about is the waste pathway. Okay, so basically what you have is the ER. It actually gets that message, just like I said before with the protein pathway. The ER gets the message from RNA to make protein. Okay, and in this case for the waste pathway specifically, we're talking about enzymes because the enzymes break down the waste. So the ER makes protein or enzymes. Now again, this protein isn't packaged the right way, or the enzymes aren't packaged the right way, so it goes to the Golgi to get packaged and distributed. But in this case, the Golgi in this particular pathway is making enzymes in a package, and those are called lysosomes. These are like mini stomachs. That's how we rub our stomach, remember, for the dance? And then what you have around the cell are vacuoles. And these vacuoles just kind of like are round, and there's like there's waste inside of it. Okay, it could be cellular waste. It could be food. Okay, whatever it is. And the vacuole just kind of goes around, and that's our vacuum cleaner motion. And what it'll do is it will fuse the lysosome, and the vacuole will fuse. Okay, so here's a lysosome, here's the vacuole, and they end up fusing. Okay, and so then the enzymes that are inside this lysosome will now break down all of this stuff. Okay, and then cells don't hold on to their waste, they actually get rid of it. So it would be sent to the cell membrane to be emptied out of the cell. The problem is that this is actually, it's going to cost energy to do this. Okay? Because getting rid of waste outside the cell membrane, that is called exocytosis. We just learned about that a few days ago. And that costs ATP, or cell energy. That is a type of active transport. Okay? So exocytosis. And one more thing, we'll talk about, what are we doing? Oh, the ATP pathway is the one that we need to do next. The ATP pathway. And it'll be our last pathway. Okay, the ATP, remember ATP is the energy currency of the cell. First thing that happens is glucose comes in to the cell through the cell membrane. It actually has to go through a channel protein to get in because glucose is pretty large, so it needs to get through that channel protein to get in. Then it travels through the cytoplasm, heads to the mitochondria, and in the mitochondria are these folds, or these things called cristae. I'll just put cristae / folds. And so what it looks like is kind of like a peanut, and then inside of it are all of these wavy lines or folds or cristae. And what that does is it allows for many glucose molecules to come into the mitochondria, like I'm talking lots, and then the glucose molecules will be converted into ATP. Okay? So glucose comes in. Okay, all these black dots are glucose, so there's many glucose coming in. And then it will just speed up and actually produce tons of ATP for the cell and to be exported out of the cell to wherever it's needed in the body. Okay? All right, and that's the end of this Throwback Thursday.